All right, here is a quick little intro to your lab that you're going to be doing today. What this is going to entail is you going through and taking a look at your lab stations. You're going to be looking at doing a lab between starch, between sugars, lipids, and proteins. Now, here are your reagents that you're going to be utilizing. The reagents are shown here. The carbohydrate test, which is for simple sugars, is Benedict solution. You're going to take a vial and put it on your desk. Carbohydrates is starch. Take a vial and put it on your desk. Some brown paper bag. You're going to take that for your paper smear test. And then finally, burette solution. You can take that to your table. Now, each group is going to get a test tube rack with 3 test tubes. Test tubes are going to be in this brown container. Label each one, test tube 1, 2, and 3 with a piece of tape. And you can grab one of these cleaners as well in between different tests that you're going to be doing. Now, we are going to be looking at testing with different reagents. And so those reagents, again, are going to be the starch, glucose, water, gelatin, and oil. And those reagents are all going to be over here on this lab table. Now, we're going to be first testing with water. Reason we're testing with water is, again, because it's a control. Starch here, make sure you mix the solution up, pour it into the beaker, and take one just eyedropper full of the material. Move your way on over to the gelatin. In the gelatin, shake it up, pour it in there, one eyedropper full in your test tube. And then over on the other side, we have glucose. Same thing, one eyedropper full is all you need. And then when you do your oil test, there's 3 drops of oil. Okay? Now, if we go back to our lab, what we have set up, I'll just show you here in a second. Lab number 1 is having you take 2 test tubes, 3 test tubes. We have our first one is going to be labeled starch, second one glucose, and the third one is water. We are testing for a starch. When we put each of the 3 liquids into the test tube, what do they look like at the beginning? I am going to add a couple drops of iodine to each of these individual test tubes. What does it look like? And if it turns black, if it does turn black, then it does contain starch. It does contain starch. Okay? Moving down to the simple sugars. We are again going to clean out our test tubes. So brand new test tubes here, washed them out. We are going to be using heat in this example. Now, this is where a Benedict solution has to be using heat. We are going to plug in the hot water bath that is at your lab station. The red light should turn on, and it should be no more about 150 degrees C. From there, you are going to just place in those 3 test tubes, again labeled 1, 2, and 3. This is a brand new set of eyedropperfuls of starch, glucose, and water. And you need approximately 20 drops of Benedict solution. Okay? After adding the Benedict solution, what color is it? Then after we heat it, that's this column. After we heat it, what does it look like? Okay? Now, only need to heat for about 2 to 3 minutes. If it changes to an orange coloration, it is positive. If it changes to, stays blue or to a green, then it's negative. Okay, move down to then the brown paper. Brown paper is the next lab. This is where you're going to be using adding a droplet of water versus a droplet of oil onto the brown paper. Let it dry, hold it up to light, see if there's any lipids in it. Same thing with oil, add 1 droplet of oil to the water, what happens? And the last one, going back to our 3 test tubes, cleaned them out. Test tube 1 is now going to be gelatin versus our starch. 2 is glucose, 3 is water. What's the color at the beginning? We're going to use burette solution, and then if it turns purple, it is positive. Remember that we do not heat this one. Okay, once you're done, answer your questions, the analysis questions 1 through 9. You do not have to do the further exploration. Okay, good luck.